In this video, I'm going to talk about mitosis and meiosis and the differences between the two. Both mitosis and meiosis are cell cycles, which is a series of events that lead to cell division. The main difference between the two is meiosis is used for the cell division of sex cells, whereas mitosis is used for every other cell in the body. The first phase of cell division is not considered a phase of mitosis, but more of a preparation phase. This phase is called interphase. During this phase, the chromosomes have replicated and are getting ready for cell division. The next phase in the cell cycle is called prophase. This is actually where mitosis begins. In this phase, sister chromatids condense, the nuclear envelope disappears, and the chromatids attach to the spindle. After prophase, cells go into metaphase. In metaphase, the sister chromatids line up in a single line along the metaphase plate. The next phase after metaphase is called anaphase. In this phase, sister chromatids separate and move to the opposite poles. The last phase of the cell cycle is called telophase and cytokinesis. In telophase, chromosomes decondense and nuclear envelope reforms. Cytokinesis is the actual separation of the cell into two daughter cells, which begins by a cleavage furrow in the animal cells. At the end of cytokinesis, we are left with two cells. Meiosis is really not too different from mitosis. Meiosis is only used in reproductive organs to make sex cells. One difference that happens in meiosis is there are two separations that occur. They are called meiosis I and meiosis II. Meiosis II is pretty much the same as mitosis, so we'll focus on the differences between meiosis I and mitosis. One difference is, in prophase of meiosis I, homologous chromosomes stick together to form a bivalent, and they switch a part of their chromosome with each other. That is called crossing over. The point of crossing over is it gives greater genetic diversity within the cells. Another difference is seen in metaphase I. Rather than lining up in a single line along the metaphase plate, the bivalents form a double line. This double line causes homologous chromosomes to separate in anaphase rather than sister chromatids separating. The cells then separate as normal in telophase and cytokinesis. After meiosis I is complete, the two daughter cells then divide in meiosis II. Meiosis II is exactly the same as mitosis. The only difference is the resulting cells. In mitosis, the result is two diploid cells, but in meiosis II, the result is 4 haploid cells. Mitosis and meiosis are obviously very important in the functioning of our bodies. Another importance of the cell division cycle is it is used to diagnose cancer, as we saw in lab. In this video, I have explained what happened in each phase of both of the cell cycles and the differences and similarity between these two.